All right, last time we finished the notes, we were talking about monohybrid crosses and how to use Punnett squares. And again, a Punnett square is just boxes that you fill in with the characteristics, the alleles. Okay, that was for a monohybrid cross, which would be just one characteristic. Well, what if you want to do two? Okay, you have two true breeding parents. And again, the F1 generation, remember, those are the kids, and then the F2 were the grandkids, and so on. But what if you want to do for two characteristics? Okay, that's called a dihybrid cross. Okay, and it's bigger. It's 4 times as much because now you got to add in all the crosses. And again, I'll show you that in a minute. But a cross between dihybrids can determine whether the characteristics are together, in other words, on the same chromosome, or are they on different chromosomes? Okay, and again, do they separate independently, or if they're on the same chromosome, they don't do that. Okay, so again, I'll show you. Here's a dihybrid cross, and you can see a monohybrid cross was this. 4 little boxes. Okay, so the P1 generation, what we're looking at here is the color being big Y, excuse me, is yellow, and then they're round. Okay, the other characteristic, the other allele would be little y's, which is actually if you have 2 little ones, you're green, so green's recessive, and then 2 r's, little r's, and those would be wrinkled. So they took the generation, we have yellow and round, and we have green and wrinkled. So again, all the children are going to be big Y, big R, little r, because that's all there is. Okay, so this is the whole generation of the children generation, the F1 generation, are going to be yellow and round. Okay, it looks like these guys disappeared. Okay, so now we can separate them, and this is a dihybrid over here. And what you have to do is, remember, in each one of your gametes, what can you have? Well, you can have a big Y and a big R. You can have a big Y and a little r. You can have a little y and a big R, or you can have a little y and a little r. You have to do all the possible combinations. So when you separate these guys into the sperm, again, you can have the 2 dominants, you can have a dominant recessive, you can have again, here's a recessive with a dominant and the 2 recessive. So you have the possible combinations. Well, the egg's the same way. Okay, so now you fill it in. All right, you get big Y, little, big R, big R, big Y, big R, little r. You just fill it in according to what's on the sides. Now what you're going to see is you're going to have 1, 2, 3, 4, 5, 6, 7, 8. I missed one. 9. Okay, so we have 9 of them that have the 2 dominant traits. Okay, now they might have the recessive trait, but again, remember their phenotype, what they look like, is going to be 9 to 3. I mean 9 for the dominant trait. Now we're going to look for some green ones. Here we have 3 green ones but they're still round, so they have 1 dominant trait and 1 recessive trait. Their color is recessive and the other one's dominant. And then here we have the dominant color, but the recessive wrinkled. Okay, 1, 2, 3. And then we only end up with 1 that has both recessive traits. Okay, this one is green and like this one way up here. Okay, and again, the only way to happen is to get both the recessive traits for the color and whether they're round or wrinkled. So your phenotype ratio is going to be 9 to 3 to 3 to 1. Okay, and again, when he did his experiments, that's pretty much what he got. So when you cross hybrids, okay, you need to do a dihybrid cross because this has 2 characteristics. If you did a trihybrid, you guys, it would have 64 boxes. Okay, because you have to put all the possible combinations. And you can do this again to figure out whether you have, if all of these stuck together, then you know they didn't separate independently. Then they'd be on the same chromosome. Okay, that's why he did it. So again, the generations, and I just went through all this, and you can see our phenotype. I'm sorry, I'm probably covering this up, but it's 9 to 3 to 3 to 1, and it's just going back and looking at the numbers here. And again, phenotype is what they look like. Okay, so a dihybrid cross, that's where he got the idea of the law of independent assortment. Okay, just because you're yellow does not mean you're going to have round cells. You could have wrinkled. So those 2 characteristics were independent of each other. They separate independently. Okay, alleles segregate independently during gamete formation. And just because you get one does not mean you're going to get the other. Okay, That's independent assortment. Like I said, that's a common with eye color and hair color. A lot of people think you have dark eyes, you're going to have dark hair. The two of them go together. I have dark hair, but I have blue eyes. That's my heritage, you know. So the eye color and hair color are not on the same chromosome. They separate independently. So you can end up with combinations. Okay. The apply only, you know, again, it only applies if they're on different chromosomes. Okay. Or if they're far apart, because sometimes you can get crossing over. But really, when it comes to it, they separate independently unless they're on the same chromosome close to each other. Okay. Maybe there's two characteristics, but honestly, I don't know an example of one. I know more of independent ones than I do of the same. Okay, so again, that's just saying just because you get one characteristic, you don't get the other one necessarily. You could, but it just depends. Okay, so if they're located on the same chromosome, they tend to be inherited together. Okay, you can have things like crossing over and stuff that can change that. All right, so so he had these two laws he came up with: segregation, which says, you know, the alleles separate during gamete formation, and they independent assortment. Again, they separate independently, and really what it does is it's probability, you know, like flipping a coin. You might get this characteristic, you might not. There's no way to really determine it until you actually see the phenotype. Okay, again, when tossing a coin, if you get heads, it doesn't mean the next one has to be heads. You can get heads or tails. So just because you get one characteristic doesn't mean you're going to get the next one. It's a toss-up. That's basically what he said. So alleles of one gene segregate into gametes independent. So you might have a gamete that has the two dominant traits. You might have one that has one dominant and one recessive. You might have one that has both recessive. They just separate independently of each other. There's no, you know, set way for them to separate. Okay, and here he was doing coins. You get the same idea: heads, tails, whatever. You can get all kinds of variants. And again, they're independent of each other. It's chance of which one you're going to get. Okay. So inheritance patterns are often more complex than predicted. Again, these were pretty simple, but again, with his pea plants, they were spot on. So again, when you get involved in higher level animals, sometimes there's more than one characteristic. Okay. With his relationship in his pea plants, it was very simple. You had genotypes and phenotypes, and they were very simple because he studied characteristics that were simple. Okay. Sometimes it's not so easy. Sometimes you have more than one gene, okay? We have a gene with more than two alleles, okay? So you can inherit A, B, or C, you know? So then you get different varieties. Another one that's complex too is sometimes there's more than one characteristic, like your height. Height in humans isn't determined like you either get a short gene or you get a tall gene. Because if that was the case, everybody would either be the same height tall or the same height short. But there's lots of characteristics that determine your height. So, you know, maybe you get 3 tall and 1 short, and, you know, you're decently tall, but you're not as tall as your parents, or something like that. So it depends on. But again, when he did his, it was green, yellow, wrinkled, smooth, you know, so it worked out really well. Okay. But many of the characteristics have more than 2 alleles. Okay. Inheritance, again, in his patterns were easy because there was just a single gene. But sometimes alleles aren't completely dominant or recessive. And that happens. Okay, it's called incomplete dominance. I think that's the next one. When incomplete dominance, again, that would be like when you have a red flower and a white flower and you cross them and you get pink flowers. That happens. Okay, so if you don't have a dominant recessive, one's not completely dominant over the other. You have that. When genes have more than 2 alleles, again, we talked about that. Your blood type, you have A, B, and O. So there's 3 there. So you have more than, you know, either not one or the other. And then you have the positive and the negative. The positive and negative, either you do it, you get it, or you don't. But your blood types can vary. Okay. And then you have genes that produce multiple phenotypes. So there's all these things that can figure into it, which is kind of crazy because none of this happened to Mendel when he picked the perfect characteristics when he was doing his work. Okay, so again, with dominance, it occurs when the phenotypes, you get a heterozygote and you get a dominant and a heterozygote, and they're not identical. So you get a hybrid basically and a homozygous dominant. Okay, you get this incomplete dominance, and like I said, that can be like a red flower and a white flower, and you get a pink flower. The crazy thing about that now, people are going to breed those pink flowers and think they're getting pink flowers, and they're not. They're going to get the red and white back. Crazy, right? But that can happen with incomplete dominance, where one characteristic doesn't totally cover up another. Okay, there's also codominant, and that's like blood types. Okay, when you get blood type, you have A, B, and O. If you get A, you're type A. If you get B, you're type B. If you get an A and a B, you're type AB. The only way you can be type O is if you don't get either of them. Crazy, right? But that can happen as well. Okay, here was like I was talking about with the red and white flowers. This is a really good case of incomplete dominance. The red doesn't totally cover it up. Okay, so you get red and white, and then you get pink. Now, if you take your pink flowers and you breed them, what ends up happening is, in order to be pink, it has to be a hybrid, a heterozygous. Okay, to be red, it has to have the two big R's. To be white, So what happens here is, if you get two reds, you're red. If you get a red and a white, you're pink. And then in order to be white, you have to get the recessive trait, okay? So when you breed your pink flowers and you think you're getting all pink, half of them will be pink. 25% of them will be white, and 25% will be red. And that's just like we did before with the other characteristics where you get when you fill in your Punnett square, okay? So the relationship between dominance and phenotype, a dominant if it's a dominant allele, it will cover up the recessive, okay? When Mendel did his, again, there was just a simple variation, but sometimes that's not the case. Okay, sometimes the nucleotide sequence allows the recessive trait to show up a little bit. Okay, and again, the more complex the organism is, the more this happens. Okay, so the alleles depend on the level, and again, you can look at the phenotype sometimes and tell, and sometimes you can't. But again, like Mendel was really, really lucky because all the traits he covered, there was no incomplete dominance. It was either the trait or not. It worked out that way for him. So when he came up with his ideas, he was spot on because he didn't have this weird variance. If he did his flowers and he had a white and a purple, and he ended up with like a light purple lavender, and he'd be going, but that didn't happen. He was lucky in that case. Okay. Some of the things that can happen, there's a disease called Tay-Sachs disease. It's very common in Jewish people, and it's fatal. And what happens is their enzyme that isn't turned off like it should be. Okay, it accumulates in their fats. And what's really awful about Tay-Sachs is the child is born and they're normal and they develop normal, and then they're about a little over a year, and then all of a sudden they go backwards. They, if they were walking and, babbling a little, they stop doing all that. And then eventually, by the time they're two, they pass away. It's a fatal disease, and there's really nothing you can do about it. And again, it's one of those, there's an allele that's recessive for Tay-Sachs, but it doesn't completely turn off. So it's incomplete dominant. And again, it's awful. It's an awful disease because, like I said, you have a normal child for about a year, and then all of a sudden you don't anymore. And then that child passes away. It's at a biochemical level, and the phenotype, because it's incomplete dominant, you totally don't shut that gene off. So it produces those fatal lipids that accumulate in the brain, and then the child ends up passing away. Okay, so at the molecular level, the alleles are codominant. All right, and again, one doesn't cover up the other, and that's what happens. But you can't see it as far as a phenotype, but you can check it at the molecular level. Okay. When you're looking at it, it's funny because you think dominant alleles, because when you combine, they cover up. You think the dominant allele would be by far the most common in populations, but it's not. And it's sometimes because isolated populations and whatever, but you get these where they're not. For example, there's a dominant characteristic where you get extra fingers and toes. It's called polydactyla, and it's a dominant trait, but 1 out of 400 people have extra fingers and toes. Okay, it's not anything that's bad. Most of the time they get them cut off because your shoes don't fit and your gloves don't fit and all that, but it's not a bad thing. It's just something that happens, and it is a dominant trait. So if it's a dominant trait, you think more people would have it. Or like brown eyes are more common, is a dominant trait over blue eyes, but there's a lot of people with blue eyes. Okay, so just because it's a dominant trait, because of all the characteristics, it doesn't necessarily going to be the one that's shown more. I know it's funny, but that's the way it happens. And we're just about out of time, so we're going to quit here, and we'll pick up next time on allele. Okay.